Okay, so now moving a little bit further into metabolism and thinking about how the TCA cycle and the electron transport chain connect. They share an enzyme in common. And this enzyme is complex 2 of the electron transport chain. And complex 2 is succinate dehydrogenase, where we take succinate, which was just made from succinyl-CoA, and it gets oxidized. But the carboxylates are not going to be oxidized. The only way that these carbons would be oxidized is if they became CO2 and left. But in this reaction, we're trying to keep all 4 carbons and work our way backwards to oxaloacetate. So what gets oxidized instead is these 2 carbons in the middle. Or there's a hydrogen that's removed. So making an alkene from an alkane is an oxidation process. But succinate dehydrogenase is the only time that we actually oxidize a carbon-carbon bond in the TCA cycle, not a carbon-oxygen bond. So the recipient of electrons, or the oxidant, is a little bit different. It's not NAD this time. It's actually enzyme-bound FAD. So oxidation of that carbon-carbon bond requires FAD, which forms FADH2. Now, this FADH2 is bound to the enzyme, or immobile. So it can't be an enzyme that's in the matrix floating around, because FADH2 would never be able to make it to the electron transport chain or the inner membrane. So this enzyme is succinate dehydrogenase, and it is bound to the inner membrane of the mitochondria on the matrix side, so that all 7 of the other TCA enzymes, which are in the matrix, can interact and trade intermediates with this enzyme. And this one can go on to participate in the electron transport chain, which is where complexes 1, 3, 4, and 5 are. So FADH2 is not a mobile recipient of electrons from complex 2. Instead, after FADH2 takes electrons, that must be reoxidized. And the mobile carrier that results from complex 2 is coenzyme Q. So enzyme-bound FADH2 is reoxidized to FAD by lipid-soluble coenzyme Q. Coenzyme Q is also known as ubiquinone. So at complex 2, coenzyme Q, which is a 2-electron carrier, becomes CoQH2, and then that is lipid soluble, which means that it can diffuse in the lipid bilayer inner membrane of the mitochondria. That can then go to complex 3, and it takes the electrons there. So this succinate dehydrogenase is sort of the most intimate connection between the matrix soluble enzymes, right? All of the other 8 in the TCA cycle, and the electron transport complexes that are permanently bound to the inner membrane. And FADH2 is not a net product, but instead coenzyme in the reduced form is the net product that leaves succinate dehydrogenase and travels through the membrane to take those 2 electrons and 2 hydrogens or I should say 2 electrons and 2 protons to complex 3. So the mobile recipient of electrons from PDC is also a compound other than FADH2, right? We said that NADH is the final recipient of electrons that result from pyruvate's oxidation. Okay, so that's another theme, right? That enzyme-bound FADH2 can't be a final product.